Hi, everybody. Welcome to another Chats with Chaudhry. I hope you're safe and well. It's a cloudy day here in Surrey today, but my day hopefully will get brightened up as I talk to my guest this morning, who is Preeti Panda, who is the Head of Business Development at Invitek Molecular. And today we are talking about sample stabilization. So good morning, Preeti. How are you?
Good morning. I'm very well, and thank you very much for inviting me for this talk.
Fantastic. And where are you calling in from today?
I am based in Berlin, Germany.
Fantastic. And is the sun shining where you are today?
We are having a summer with a bit of rain.
All right. Well, hopefully it'll be sunny later on today, as I hope it starts to shine here as well in the UK.
Hopeful.
So before we start talking about sample stabilization, would you mind giving viewers a quick overview of Invitek and also your own background?
Sure. I'll be pleased. Invitek Molecular is a German company who offers tailor-made nucleic acid purification and sample stabilization solutions. They're here since 2 decades with wide expertise of various sample materials from human, animal, food, environment, forensics, and henceforth, basically one of the pioneers in pre-analytical stage of biological sample management, offering both off-the-shelf and custom solutions to our partners.
Great.
Coming for me, I'm from India. After completed my master's in biotechnology, I have been working for about 8 years in the instrumentation industry, catering molecular research and diagnostics. Worked in multiple roles from technical to sales. Currently, I'm leading the business development at Invitek as a manufacturer and solution provider in the IVD product domain.
Brilliant. So let's talk about sample stabilization. For someone who is a layperson like me, really basic question. What do we mean when we say the word sample stabilization, and why is it important?
Sample stabilization means stability of a genetic material to process any molecular downstream analysis. Why is it needed? It is essential because if a biospecimen experiences any change between collection and downstream analysis, the sample may not yield enough quality DNA to conduct the analysis. Right. If the data is generated and analyzed from these changed biospecimens, we will not be procuring the true representation of the samples, which will lead us to flawed and irreproducible results.
Right, okay. And what are the main industries that sample stabilization used in?
Sorry, can you repeat the question?
What are the main industries then that sample stabilization is used in?
So sample stabilization, or if I term as sample collection devices, which includes the DNA stabilization buffers, are opening new possibilities for home-based or self-testing, which is basically implemented in multiple DNA testing services, such as ancestry screening, microbiome profiling, nutrition chart screening, allergy screening, etc.
Right.
So to put it in a simple note, it's basically a simple and compact solution for enabling efficient operations for any lab or service diagnostic company by creating efficiency in the testing services. Not just that but also a good high quality standard experience for the patients right at their doorsteps.
Right, okay. So let's look at some of the challenges. What are the main challenges then within sample stabilization when people are trying to take a sample and use it?
So sample stabilization is a crucial first step in any molecular assay. But the instability of the molecular profile, which degrades very rapidly after collection of the sample, poses a major challenge. Hence, immediate stabilization is recommended for ensuring the reliable interpretation. The possible challenges with instable samples for instability of the samples are fluctuations in temperature. Increase in temperature will quickly damage and degrade the DNA. The longer the sample is unstabilized, the greater is the likelihood for the sample or the specimen to change as a result of environmental impact and also natural biological degradation. Other possible factors as a challenge for sample instability is irreproducible samples or inhomogeneous mixtures, integrity and intactness of the DNA, which is affected with any fluctuation, the turnaround time of the sample to reach the laboratory, and henceforth. So, yeah.
Right. Okay. And so how do you overcome these particular challenges then?
To obtain, I would say, to obtain truly accurate outcome, a sample must be stabilized and protected from the point of collection until it is analyzed downstream. So the current approach which is being recommended now is with non-invasive sample collection, which is most conveniently with saliva, swabs, stools, etc. It is easy to be used by everyone and also for shipping at room temperature without any cold chain requirement, which saves costs and also avoids the sample degradation during transportation or until the extraction has been conducted. Here, Invitek has developed sample stabilization solutions for sample types such as stool, swab, and saliva, which are easy to be transported, ensuring the stability and integrity of the DNA samples to be intact.
Right. Okay. So what makes your services unique then and your technology different?
So Invitek has patents and has a proprietary buffer composition, which enables the samples to be preserved for a longer shelf life at room temperature without any requirement of the cold chain. The traditional concept lies with as simple as lysis of the cell walls, or the first step lies with the lysis of the cell walls, which makes the DNA to be released in the controlled environment of sample stability, ensuring a longer duration of the samples to be stable in the required buffer.
Right.
I would say that I would certainly like to introduce a couple of products which we have. For stability, these are the handy packs which many of our providers off the shelf or as custom solutions order from us. These are in the packs. Maybe I will introduce it like this. This is a stool collection device. It includes a liquid buffer with all barcode information to enable proper identification of the sample. It has a spoon-based structure to ease of collection, and once the sample is collected, it ensures stability up to 3 months. The other technology which I would certainly like to introduce is the saliva gene collector, which is a collection preservation device for saliva using a lyophilized buffer, which basically gives the advantage of non-leakage or leakage proof, because it's not a liquid-based buffer, it's a lyophilized buffer. Once the saliva is collected as per the required volume, the solution dissolves in the samples and enables the DNA to be preserved for up to 1 year.
Right. Fantastic. So what are some of the more common questions in that your customers ask you then around sample collection, sample stabilization?
Sure. So many of our partners who are basically laboratory service providers, they're curious to understand about the compatibility of the collection device to the downstream applications. And I'm happy to say that the compatibility, the stabilization units are compatible to all the molecular assays, regardless of which applications you're opting for. The main concept behind, the objective behind the stabilization is to preserve the DNA in a proper environment and not let it degrade.
Right.
The other common questions which we found is how long can you preserve the samples? And here the answer would be it depends upon the sample types. The stool up to three months once the sample is collected. And with saliva samples, our units could stabilize the sample for up to 1 year.
Right. Okay. Now, obviously, looking ahead, I mean, when you look at a lot of diagnostic equipment and also at home testing, you're getting a lot of, particularly within the diabetes field, for example, the use of AI and smart technology, where data is collected from the patient at point of use, and then is collected by GP and researchers or whatever. Now, do you think that will be the future for sample collection as well, in terms of people be able to, when they take the sample, be able to diagnose it and collect that data there and then, rather than having to transport it to the lab? Or do you think that's possible now, or is that something for the future?
I think that your question is pointing more towards the point of care testing. When you refer to the diabetes application, which we are currently using with
Sure.
monitoring systems, I would say yes. For immediate answer to go ahead with a particular treatment, yes. But however, for a detailed analysis, I would say that we still depend upon the golden standard of molecular assays such as qPCR and NGS.
Right, fantastic. All right, well, thank you for that. So looking at, well, final question, where can people get more information?
Okay, for all the solutions offered by Invitek, we have a very friendly website, invitek-molecular.com. Please visit that. You can find all the video tutorials of how the samples are collected, the usefulness, the key features on our LinkedIn website as well. We upload all the videos there, and we are also soon going to upload it on our website, or it's on progress. For technical information, please visit the products domain in our website, where you can find the brochures and white papers related to specific product domains such as microbiome, infectious diseases, genetic testing, life sciences, etc.
Fantastic. Well, Preeti, I know that's all we've got time for. So I hope the sun shines for you today in Berlin and you have a lovely summer. So thank you very much for the time.
Thank you so much, Rizwan.
Thank you. And viewers, I hope you found that useful. If you'd like to know more about sample stabilization and the collection of samples, as well as the services Invitek Molecular offer, then please check out the links above the video. They're all there, so you'll be able to check out the videos on their LinkedIn page, the application notes, the white papers, and other information as well. So all this to say is Preeti, thank you very much once again.
Thank you.
Enjoy the rest of the summer, and thank you, viewers, for watching. I hope you found that useful. Until next time, as always, stay well and stay safe. Bye-bye.
Bye-bye.